Hello, my name is michelle-2. The topic of the day is epigenetic control of interleucin 9 in asthma. Definition: interleucin is a pleiotropic cytokine associated with allergic inflammation and is therapeutic target in patients with asthma. Its expression and that of the interleukin receptor are agreed in the lungs of persons with asthma that is those of healthy persons.
Here's a conundrum. Identical twins originate from the same DNA, so how can they turn out so different, even in traits that have a significant genetic component? For instance, why might one twin get heart disease at 55 while her sister runs marathons in perfect health? Nature versus nurture has a lot to do with it, but a deeper related answer can be found within something called epigenetics. That's the study of how DNA interacts with the multitude of smaller molecules found within cells, which can activate and deactivate genes. If you think of DNA as a recipe book, those molecules are largely what determine what gets cooked when. They aren't making any conscious choices themselves. Rather, their presence and concentration within cells makes the difference. So how does that work? Genes in DNA are expressed when they're read and transcribed into RNA, which is translated into proteins by structures called ribosomes. And proteins are much of what determines a cell's characteristics and function. Epigenetic changes can boost or interfere with the transcription of specific genes. The most common way interference happens is that DNA, or the proteins it's wrapped around, gets labeled with small chemical tags. The set of all of the chemical tags that are attached to the genome of a given cell is called the epigenome. Some of these, like a methyl group, inhibit gene expression by derailing the cellular transcription machinery or causing the DNA to coil more tightly, making it inaccessible. The gene is still there, but it's silent. Boosting transcription is essentially the opposite. Some chemical tags will unwind the DNA, making it easier to transcribe, which ramps up production of the associated protein. Epigenetic changes can survive cell division, which means that they could affect an organism for its entire life. Sometimes that's a good thing. Epigenetic changes are part of normal development. The cells in an embryo start with one master genome. As the cells divide, some genes are activated and others inhibited. Over time, through this epigenetic reprogramming, some cells develop into heart cells and others into liver cells. Each of the approximately 200 cell types in your body has essentially the same genome, but its own distinct epigenome. The epigenome also mediates a lifelong dialogue between genes and the environment. The chemical tags that turn genes on and off can be influenced by factors including diet, chemical exposure, and medication. The resulting epigenetic changes can eventually lead to disease if, for example, they turn off a gene that makes a tumor-suppressing protein. Environmentally induced epigenetic changes are part of the reason why genetically identical twins can grow up to have very different lives. As twins get older, their epigenomes diverge, affecting the way they age and their susceptibility to disease. Even social experiences can cause epigenetic changes. In one famous experiment, when mother rats weren't attentive enough to their pups, genes in the babies that help them manage stress were methylated and turned off. And it might not stop with that generation. Most epigenetic marks are erased when egg and sperm cells are formed. But now, researchers think that some of those imprints survive, passing those epigenetic traits on to the next generation. Your mother's or your father's experiences as a child or choices as adults could actually shape your own epigenome. But even though epigenetic changes are sticky, they're not necessarily permanent. A balanced lifestyle that includes a healthy diet, exercise, and avoiding exposure to contaminants may, in the long run, create a healthy epigenome. It's an exciting time to be studying this. Scientists are just beginning to understand how epigenetics could explain mechanisms of human development and aging, as well as the origins of cancer, heart disease, mental illness, addiction, and many other conditions. Meanwhile, new genome editing techniques are making it much easier to identify which epigenetic changes really matter for health and disease. Once we understand how our epigenome influences us, we might be able to influence it too.
Application in medicine. Epigenetic modification play an important role in the regulation of different cellular functions, including inflammatory allergy response, DNA repair, and cellular proliferation, differentiation. Several environmental factors, for example, exposure to allergens, tobacco, bacteria, microbial components, diet, obesity, and stress, have been shown to have an epigenetic influence from the intrauterine period on the genetics of allergic disease and asthma.
Epigenetic modification of the epigenome is established during early embryonic development and is maintained during cell division. In this video, we will look at DNA methylation during embryonic development and the influence the environment can have on DNA methylation patterns. During the period of embryonic development, methylation patterns of the germline and somatic cell lineages are established. Here is a pregnant woman, and we will zoom into the uterus, and from here go back in time to when fertilization occurred. Then we will follow the changes in DNA methylation pattern during embryonic development. Initially, the egg that is released during ovulation carries the maternal DNA methylation pattern. The sperm that will fertilize the egg carries the paternal DNA methylation pattern. It is important to draw a graph. Here we have on the y-axis the level of DNA methylation pattern, and it can be increasing or decreasing during early embryonic development. The female egg and male sperm, as I already mentioned, carries its own DNA methylation pattern. During fertilization, a zygote is formed, and the maternal and paternal genome and epigenome are contained in one cell. The paternal epigenome then rapidly undergoes genome-wide active DNA demethylation and remains demethylated following multiple rounds of cell division. The maternal epigenome undergoes gradual, passive demethylation. By the morula stage, the methylation pattern of the embryo is essentially erased. At the blastocyst stage, de novo methylation is initiated. De novo methylation is where new methylation patterns are re-established in the growing embryo. During differentiation, each cell type acquires a unique DNA methylation pattern that is subsequently maintained during each cell division. The initial enzyme responsible for removing DNA methylation patterns from the maternal and paternal genomes are enzymes called TET. Methylation are carried out by DNMT enzymes. DNMT3A and DNMT3B are responsible for de novo DNA methylation of embryonic cells. And DNMT1 is responsible for maintaining the DNA methylation state in somatic cells during each cell division. DNA methylation plays an important role in human health and has been associated with a growing number of diseases including cancer, imprinting disorders, and repeat instability diseases. Although the DNA methylation patterns are relatively stable, changes in methylation can potentially occur during early development due to environmental influences. For example, the mother's environmental exposure to smoke, alcohol, nutrition, and drugs may lead to random epi-mutations in the developing embryo, and even in the germ cells of the developing embryo. The developmental origins of health and disease hypothesis proposes not only that we are what we eat, but also that we could be what our parents and grandparents ate and what they have been exposed to. The epigenome, unlike the genome, can also be modified in response to interactions with environmental conditions after birth. And accumulated DNA methylation alterations as we age can lead to diseases such as cancer.
Pros and cons. The pros sound that this modification are potentially reversed and could be targets for the development of future therapies include asthma or allergy. Cons, the role of the different mechanisms of epigenetic regulation present has yet been clarified. Clinical studies are needed to clarify the exact mechanism of epigenetic modification and the role in the genesis of allergy disease and asthma. Issues applied in Ecuador. The article points out the relevance of the environment conditions and the type of exposures to stimuli as regulators of gene expression through a specific epigenetic mechanism. It's an interesting study, but nowadays is not applicable in Ecuador. The conclusions is describe a new layer of epigenetic regulation in the production of asthma-associated cytokines and have therapy provide another point to target dysregulated immunity in patients with asthma or allergy disease. Because asthmatic low disease has long been associated with increments expression of histone acetylase and reduced expression of histone deacetylase. It is tempting to speculate that CD4 T cells in patients with asthma may show agreement superenhancer activity at the interleukin-9 locus and other loci, sequence to environment or genetic influence, or a combination of the two, and that such activity could potentially explain the predisposition of the development of atopic disease in some, but not all persons.
Hello, my name's Marnie Blewett, and I'm a laboratory head at the Walter and Eliza Hall Institute of Medical Research. And I have a lab that works on epigenetic control, and I teach epigenetics at the University of Melbourne. So this course on epigenetic control of gene expression is going to be a really exciting course. So what is epigenetic control? One of the things that really fascinates biologists is how it is that all of our different cells within a human body can have the same genetic information, the same book of instructions, and yet end up with such different functions. So if you compare the function of a cell that is found in a hair follicle and it's involved in making keratin and making the hair shaft, compared with a liver cell, they have distinctly different functions, and yet they have the same set of instructions. So what we now understand is that this is enabled by having only some of those genes or some of those instructions being used at any one time in each of these different cell types that we have in our body. So how is it that we can have some genes being used or switched on and some genes being switched off? And really this happens by epigenetic control. And what this course is about is teaching you about how epigenetic control works within the cell at the molecular level and how that leads to different expression of different genes within each cell type. The simplest way to think about epigenetic marks or epigenetic control is like punctuation marks in English. So if you think of the string of 26 letters that we have in the alphabet, if you put them all together and you remove all the commas and full stops, capital letters, spaces, exclamation marks, they're just strings of letters, and it's very difficult to see what a sentence might say, let alone a paragraph or a chapter or a whole book. Well, in the human genome or any other genome, it's the same sort of thing, except that here we just have 4 letters rather than 26. And epigenetic marks are a bit like punctuation marks. They like that formatting, the exclamation marks, the full stops and commas. They allow the cell to be able to see each gene and see each piece of information. They allow them to read it. So, for example, exclamation marks might be on genes that need to be used within a particular cell, and full stops might be on the genes that don't need to be used. And so if you keep this analogy of punctuation marks in mind when you learn about epigenetic control, it'd be very helpful. So what this course will do is go through some examples of how epigenetic control works at the molecular level, how what we have learned from lower organisms like yeast, plants, flies and worms, and then move on to the majority of the course, which will be focused on mammalian epigenetics. So how epigenetic control contributes to normal development and how it goes wrong in disease. So I hope you'll join me on this exciting learning of epigenetic control. Thanks.
Thanks.